Hello, it's Étienne Danchin from Toulouse in southwestern France, and I'm giving lecture 8 about culture and insects. And the lecture will have two parts. The first one will be we'll end up with a new definition of animal culture, and in the second part I will develop a specific example. Several points of introduction. First, a brief history of animal culture. Until the end of the 1980s, culture was mainly human-centered, meaning that definitions were designed to capture the originalities of animal culture, of human culture, and because of that, they were inappropriate to study animal culture. It was also mainly theoretical, with very few experiments, but in effect, the history of the study of animal culture emerged from the field of human sciences. From the early 1990s, the field of animal culture emerged, and mainly through the study of animal social learning, and you know a lot about it because you've got two lectures, already had two lectures about this. It was mainly in invertebrates at the beginning, but after the end, let's say at the moment of the end of the 2000s, the field of animal culture emerged in insects with a few examples. We'll see this later on. And again, at the end of the years 2000s, it also was included into the field of behavioral ecology, which is a study of the evolution of behavior, with, for instance, a full chapter about animal culture in a textbook that I've been writing with several colleagues. And in parallel, at the same period, more experiments started to emerge from this field. And in fact, there were three main approaches during this long history of research. The first one was modeling, coming from human sciences, and the goal was to study the conditions of emergence and the consequences, the potential impact of animal culture, of human culture, on evolutionary and ecological processes. The second method was the study of animal social learning, and you've had several lectures about this. You know quite a lot about it now. And the third point, you know also a little bit, quite significantly about it, was about reporting the existence of persistent patterns of behavioral variation across populations that we call traditions. In this population, they behave one way. In another population, they behave differently. And this is persistent in time, and all the individuals of the same population are homogeneously behaving the same way. And these three approaches produce the wealth of data that are presented in this module. But as every approach, there are limitations, there are pros and cons, and in the cons aspect, there is the fact that there were very few experiments in effect, which didn't allow us to study the causality of the processes. Also, there is an important point that I'll come back on later on, which is that social learning in itself is necessary, definitely necessary, there is no doubt about that, but it is not necessarily sufficient to generate a cultural process, and we'll see why and how. The third point I wanted to highlight here in terms of limitations is the fact that the approach of traditions, which has been extremely productive and very interesting because it's settled the field, in this approach, it's really hard to rule out other explanations than the one that could explain the patterns than social learning. For instance, it could be ecological differences. Maybe in one population they break nuts with stones because there are stones here. In another population there are no stones for some reasons, and then they use a stick, for instance. And it's very hard to rule out all these potential possibilities. It is even harder with genetics because if we want to be ultimate, you would need to take every single base pair within the genome of a species to test whether there is no variation in this single base pair that is associated with behavioral patterns. And it's almost impossible to do. So clearly we needed to develop new approaches. And this is one of the things I would like to highlight while talking about the case of insect culture. And what is behind this is a question about the link between patterns on the one hand and mechanisms on the other one. It is clear that the same pattern, here traditions, can be produced by many different processes, many different mechanisms. So if we want to be sure that the pattern we observe is produced by social learning characteristics, we need to focus on mechanisms, and particularly on the properties of social learning. And this implied somehow to rethink the definition of animal culture. And this is what I will do towards the end of this first part of the lecture. Before, I'd like to show you a brief review of what we know about social learning in insects. Does it exist, and to what extent is it present? And I will give evidence coming from three fields, three kinds of contexts in insect social learning. The first one is about foraging, with two examples. The second one is about danger, with one example. And the third one is about mate choice. In the context of mate choice, do animals use social information? So in the context of foraging, the first example is the case of the bee dance. It's the first and most famous example that was discovered by Carl von Frisch, who got a Nobel Prize for this fantastic discovery. He found that in the hive, the bees on the walls that are vertical performs a dance that we call the bee dance that consists in a straight line, a vibrating straight line like this, that has a specific direction on the vertical plan, and then it goes back, and then dances again, and back, and so on. And he showed that the angle of this straight line relative to the vertical which represents the direction of the sun, represents the angle, this alpha here, the angle of the direction a forager should go to find the new food source that has been discovered by this forager, specific forager. So clearly, the bees are indicating the direction in which a forager should go to find this new source. And the intensity of the vibration here, the characteristics of the vibration here, will indicate the profitability of this food source, saying something that it is that direction about 500 meters, let's say. So clearly it was a fantastic discovery, and Carl von Frisch was able to be at the hive, not knowing where his colleagues would put the food source, and would be able to guess where it was. It was a really convincing experiment. The second example in the context of foraging is still with bees, but bumblebees this time. They are pollinators. Most of that species, Bombus terrestris, is a pollinator, and they feed on the nectar produced by flowers. And in doing this, they pollinate the plants. But there are cheating plants that look very similar to nectar-producing flowers, but that do not provide nectar. So they are fertilized by naive bumblebees that believe that they will find some nectar. And of course, there is a strong selective pressure for bumblebees to be able to detect who is a cheater, who is a nectar-producing flower. So a student performed an experiment in which there were two kinds of flowers that were slightly different, very similar, but slightly different, and detectably from a bee point of view. So there was a dark and a light flower, and randomly one of these colors were attributed to being a provider and the other one being a cheater. And she built a field like this with flowers that could be either aggregated by kinds or mixed together. She studied this as well. And in this field, she would release a naive bumblebee that would be either alone, and she would have to learn on its own, or with a guide is in the know, that knows already, that has already been trained in the system, and who knows that the dark one, for instance, in this example, have got nectar, while the light ones are cheaters. And she found that this was efficient, because if you've got here the rank of the visits, the first flower to be visited up to the 20th to be visited in the system, she found that the ones that are naive are performing really badly at the beginning, while after a certain amount of experience, they in the end raise the same performance as the ones that started being naive but with a guide. These ones start very high immediately because they've got a guide and they tend to follow them and to go on the same kind of flower as them, so they are efficient rapidly. You might think that this is not a big difference, but in fact, if you think in terms of an ecological context, there are many foragers, and a bee would have a big advantage in being the first to get into a given flower, because otherwise, if it goes to that flower a little bit later, it might have been emptied by someone else before. So the impact on the feeding efficiency can be quite significant, and thus on fitness. So clearly, it showed that in the context of foraging, we have some really good evidence that bumblebees and bees in general can learn from others. In the context of danger, it's a story of crickets and spiders. There were true treatment in this experiment. Another student who did this, and the experiment was this. You had two tanks, well, repeated with replicates, of course. In the danger situation, you had crickets, 10 crickets with leaves, dead leaves at the bottom, so that they could hide in this tank. And in one of the tanks, you just added 3 spiders, danger. And the other one, you add nothing else. So when you do this for 2 days, after 2 days, you can observe the crickets, and you can realize that there is a big difference, a significant difference between the crickets in this treatment and in that treatment. These ones are hidden. You can't see them. They are hidden. They're hiding from the spiders, and as a consequence, they are hiding from us as well. So clearly, there was a big difference, a big effect of the presence of spiders. A way to deal with the danger was to hide. Then, after 2 days, she transferred the crickets from the 2 types of tanks to a new tank, and this time without any difference, without spider. So no more spider in the system. And she added on each of these 5 new crickets that are completely naive, that don't know anything about what happened before, and they stayed together for 6 hours. It's a relatively short period of time. Then, after 6 hours of being together, she removed the demonstrator crickets, the one that were confronted to spiders, and the same thing here. And she lets the naive crickets to behave and to be in the tank for 24 hours. And at that time, she now measured again the visibility of the crickets. And she found again a big significant difference between these 2 treatments. The one that were in this treatment were much more hidden than the one that were in this treatment. These ones had somehow learned from the behavior of the demonstrators that there was danger in the environment. So clearly, in the context of danger, you can learn a lot of things from your conspecifics somehow, and insects can do this. In the context of mate choice now, it's a completely different context that has been relatively rarely, well, there is a big literature about mate copying, which is about mate choice, and the question was to study this. And we did, this experiment here was performed in Drosophila melanogaster, which is a really minute fly. And the experimenters were able to create 2 new categories of flies, of males, some that were dusted in green and some that were dusted in pink. So 2 new male phenotypes, and you can see that in this example, the green male was somehow selected by a female to copulate with, while this pink male was apparently non-selected, so we can interpret it as it was rejected somehow by this female. So this copulating pair here constitute a demonstration, as the crickets that were scared by spiders were demonstrators in another context. And interestingly, you can see that there are 2 females here that seems to be eager to watch what is happening. Aha, Mr. Green is copulating with a female, while Mr. Pink wasn't selected for that. So now imagine that we test these females that observe this and discover that they love green because of this, what they've seen. Then you would say, well, some social learning can exist. So we transferred this situation to the lab with a system, a device like this one, in which you've got two tubes facing each other, separated by a glass partition, which is either transparent, they can see each other, or opaque. On one side, you put the observer female, the pupil, the one you want to teach something to. On the other side, you give a lecture, which you call demonstration, which consists of a female that is choosing around freely between a green and a pink male. And imagine that she will start copulating with a pink male. After this demonstration that lasts about half an hour, they removed all the flies on this side and replaced them by one new green male and one new pink male. Okay, one of each. They removed the glass partition, and they recorded with whom this observer female now copulated. And this is a mate choice test in which, in this example, is Mr. Pink who was successful, while Mr. Green didn't get copulation. Now, what about the results? They found that when they are uninformed, that is the same design, but the glass partition was opaque, so that the observer female couldn't see what was happening in the other compartment, then they behave randomly. They choose green or pink randomly. On average, it's 50-50. But when the glass partition was transparent, meaning that the observer female could see what the choice of the other female, now they found a strong, a significant preference for the male of the color that was selected during the just previous demonstration. So it was clearly an example of speed learning, speed social learning, speed because it was only one demonstration that triggered this significant and efficient social learning. And so females showed now, that observed the copulation, show a bias for the males of the phenotype that was accepted over the males of the phenotype that was rejected during the previous demonstration. So now we know a little bit about the evidence that exists for social learning in insects. What about the evidence that exists for insect culture? And it shows there are very few examples. There are some interesting indications, but culture, the only really, one of the strongest examples I can think of was the string pulling in bumblebees. The experiment was very simple. It was again using artificial flowers. Here a blue flower in which you put some sucrose, and bees can easily learn that this blue means some food, and they start feeding on these flowers. And then, step by step, you start hiding this flower under a transparent table so that it becomes impossible to feed on it. And here, clearly, the bumblebees cannot reach the food source and can't feed. But you attach a little string to this flower when you hide it behind the table, and rapidly, step by step, the bees learn to pull the string in order to move the flower outside of the table, and then they start feeding a bit in a situation a bit like this. And you can see that they can learn it relatively rapidly, and their performance increases a lot. That is, the time they spend to get the reward goes from something like 250 to something that is less than 25. So the time is divided by more than 10 times. 10, it's quite a big improvement. And then once you've got a bee that is trained like this, they introduced it into its own colony, and they looked at the way the string pulling performance spread within the colony. And here are the results. You can see that you've got the different members of the colony here. They are all tagged, so you can monitor each of them. Here is the individual that was trained before, that was introduced into the colony after, and you can see that it has got connections with other individuals. And one of these individuals in connection started to adopt this behavior as well, and this one was copied by many other individuals. And after 4 days, you can see here, after 4 days, most of the colony was able to pull the string to get the food. So it's a fantastic example of social learning, of transmission within a colony, which is extremely impressive, and I like this experiment a lot. But there is a but. It's often like this. We can always improve things. And my problem is that it is only transmitted within the colony. This experiment doesn't show and didn't study whether it is also transmitted from one colony to another one. And this would be really important because if we want this habit of string pulling to persist, it would need to be transmitted across colonies because we know colonies are kind of individuals, meta-individuals, that will die at some stage. And if they die without having transmitted, this habit will disappear. So it's not completely convincing that it is an example of culture. It's a fascinating example of social transmission and social invasion somehow. But I'm not definitely sure that it can be qualified of what is that it is an animal culture example. But this just slide is just about raising the question about what do we mean exactly by animal culture. And this will be the last part of this first section of this lecture 8. What is animal culture? And we want to answer this in the light of what we now know, because you've listened to several lectures about social learning in particular, and the history of the field of animal culture. Historically, it is in the 1990s about this mid-copying, this social learning in the context of mate choice that emerged this discussion. Also started to underline the fact that social learning in itself is not sufficient to prove the existence of culture. We need more information. It is absolutely basic and necessary, but it's not sufficient. So we said at some stage, it is the first criterion, it's the basic one, without which nothing else is interesting. It can be asked. But once you've got this, you can also ask about the properties of this social learning. And it should be, in particular, it should be transmitted from slightly older to slightly younger individuals in order to, for this behavior to be imitated, copied, learned by someone else in the population. So it's criterion 2 of transmission across age classes. If it is only horizontal, it will be fascinating, but it will disappear with a cohort of individuals. It should also be memorized for sufficient time for the individual to perform it and thus to be possibly copied by others. This is criterion 3 of durability. And the fourth one is a bit subtle, but it's really important because in the mate copying example I showed you, the social learning in the context of mate choice, the female in Drosophila might learn to prefer one male over the other one, which is interesting in itself, but it's not sufficient because these two males and the next generation will be dead. So if all of the females start to learn to prefer that male over that male, this is interesting, but it won't generate a tradition, a process of cultural process. To do this, the females have to learn to prefer a given trait, not a given individual, but a given trait. Here, green versus pink, or vice versa. So it should be trait rather than individual-based. And these are the 4 criteria that we had detected in 2010 when we published this with Richard Wagner, and we wanted to integrate it into a single inclusive definition that we would have a pattern part and a mechanism part. The first part is pattern is about we formulate it that way. This is the part of phenotypic variation that is transmitted across generations through social learning. In other words, it's socially inherited variation among populations, and it's more centered on patterns of traditions. But still, already we say that it's socially inherited, so clearly we pinpoint a mechanism of transmission. And to study this mechanism of transmission, it is important to study the properties of social learning and whether they can lead to patterns of cultural tradition. In this behavior, females tend to prefer, for instance, green, and in another population, they will prefer pink, while there is no fitness effect of this in the first step. And why cultural tradition? Because this is the most striking marker of culture. And again, it's focusing more on the mechanisms, how animals learn and does this way of learning has the potential to generate cultural traditions. So we had 4 criteria, and we said in 2010, let's be demanding. Let's ask for the 4 criteria to be met simultaneously in the same system to be able to claim that the trait is at least partly culturally transmitted. And the next step was obvious. It was to apply it to a single system this definition by testing these 4+1 criteria of that system. And the +1 is very important, you'll see in the second part of this lecture, because at that time, in 2010, we had missed a really important criterion, which is now criterion 5, and I will see this in the next part of the lecture.